Say that you just had a meal. So you have three biomolecules here. So it could be carbs or lipids or proteins. In this case, say that you just had a hamburger. And now you're going to go through glycolysis. So very first of all, you're going to have a lot of glucose molecules in your bloodstream. So all those macromolecules are going to get broken down into very small biomolecules. So in this case, you have glucose molecules. And when you have a lot of glucose molecules in your bloodstream, what's going to happen to your blood level? So you're going to have increased blood sugar level in your blood, right? So when you have increased blood sugar level, then what happens? You're going to activate the pancreas. So now your pancreas is very happy because now it's going to have time to work. So pancreas get activated. So what happens if the pancreas get activated? Then it's going to produce a lot of insulin molecules. What happens when you increase the insulin level? Then all those insulin molecules are going to go to the receptor, its receptor, insulin receptor, and it's going to activate the receptor, and that in turn is going to activate the GLUT transporter. So this is a transmembrane transporter. So this is going to bring the glucose from the bloodstream into the cell. So now you're going to go through the plasma membrane. So that's the PM, plasma membrane. And now the glucose molecules are inside of the cell. So now it's within the cytoplasm. Now we have to capture these glucose molecules so it doesn't diffuse out through the plasma membrane. So what do? You want to phosphorylate that glucose molecule so that it's trapped inside of the plasma membrane, so it's within the cell. So what's the enzyme you use to phosphorylate a molecule? It's a kinase. For the muscle, we'll say hexokinase, and the liver, we say glucokinase. And when you dephosphorylate, the enzyme would be phosphatase. Okay, so now it's within the cell and it's trapped, so it's not going to diffuse out from the cell membrane, and it's going to go all the way to the last molecule using the glycolysis pathway. We're gonna start with glucose 6-phosphate and change the form into fructose 6-phosphate. So why do we have to keep changing the figures here? Because we want to eventually break that 6 carbon molecules into 3 carbons and then keep breaking down so that we could have a final energy. And what's the enzyme that we're going to use to change the form of glucose to fructose would be isomerase. And we're going to attach another phosphate group. Again, what are we using here? It's another kinase. So it's going to attach on the first and the sixth carbon of the fructose. So we call that fructose 1,6-bisphosphate. In this case, it's not called diphosphate because they are not right next to each other, but those phosphate groups are from far away from each other, right? For the first and the sixth carbons of the molecule. And using the kinase. Adding the phosphate, kinase. Removing the phosphate, phosphatase. Now we're gonna finally cut the molecule in half. So now we're gonna have 3 carbon molecules. So we have dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The molecule is broken down by aldolase. So this pathway goes a little bit fast or faster than the other regulation sites because this is not the regulation sites. So it's going to produce energies from now on, and the DHAP molecule will convert into glyceraldehyde 3-phosphate pretty fast. So instead of DHAP, we're going to use glyceraldehyde 3-phosphate, and it's going to convert really fast. So now we're going to have 2 molecules of glyceraldehyde 3-phosphate instead of 1 glyceraldehyde 3-phosphate. So just be aware that in the textbooks or some papers, we might not see that 2 for every molecule that's in the pathway, but just know that from now on, there are 2 molecules for everything that we produce. So we're going to have 2 1,3-bisphosphoglycerate, and we're gonna produce 2 ATPs, 2 NADH, so all the way down to pyruvate. We're gonna have 2 molecules for each of them, starting from 2 molecules of glyceraldehyde 3-phosphate. How do we get to 3-phosphoglycerate? Because we just got the phosphate group removed, producing ATP from the first carbon. So we have 3-phosphoglycerate, and using a mutase enzyme, we change that to 2-phosphoglycerate. So we have 2,2-phosphoglycerate, right? So from this 2-phosphoglycerate, you are going to produce a byproduct, water molecules, which is not a lot, but it's going to balance our system, and also phosphoenolpyruvate, PEP. And finally, we have the last product of this pathway, pyruvate. So we have the 2 molecules of pyruvate from this glycolysis pathway. So using this pyruvate, there are many ways this could go into in metabolism. So we call the fate of pyruvate. So what can we do with this pyruvate? First, when you have oxygen, which is aerobic, so the pyruvate will go into mitochondria. So once it goes into mitochondria, it's going to go through the Krebs cycle, and it's going to produce a lot of energy from the Krebs cycle. 2, when there's no oxygen involved, which is anaerobic, pyruvate will go through lactic acid fermentation, which will produce lactate, so such as in the muscles. And number 3, it could also go through gluconeogenesis. We'll talk more about this gluconeogenesis pathway after we have finished the Krebs cycle and all different cycles that's involved in the metabolism, because this is pretty specific. And number 4, it's going to produce or synthesize amino acids. So you're gonna have protein metabolism later. But for now, pyruvate will produce amino acids such as alanine. Now that you know glycolysis, can you teach others using the same figures?